Today, we'll be examining the molecular mechanisms that allow the bowhead whale to live for over 2 centuries with a low incidence of cancer. This analysis, based on the work of Firsanov and colleagues, explores the genetic underpinnings of this remarkable longevity. We'll find that their primary strategy may not be suppressing tumors after they form, but rather preventing the initial genetic damage that leads to them.
The observation that bowhead whales have an exceptionally long lifespan is not new. In fact, it's rooted in the traditional ecological knowledge of the Inupiat people of Alaska. This long-standing knowledge provided a crucial impetus for scientific investigation into how such a large mammal can sustain itself for more than 200 years, and what genetic adaptations make this possible.
Here is the structure for our analysis. We will begin with the central biological problem, known as Peto's paradox. Then, we'll review how conventional hypotheses for cancer resistance were tested and ruled out. This leads to a counterintuitive finding about the number of oncogenic hits required for transformation. From there, we'll delve into the core mechanism, enhanced DNA double-strand repair. We'll identify a specific protein, CIRBP, that facilitates this process, and conclude by discussing the broader implication of these findings for longevity and health. Let's begin by defining the core biological question that this research addresses. The extreme size and lifespan of the bowhead whale present a significant challenge to our understanding of cancer biology, a challenge known as Peto's paradox. Peto's paradox describes the observation that large, long-lived animals do not have a higher incidence of cancer compared to smaller, shorter-lived ones. From a probabilistic standpoint, this is unexpected. Every cell division carries a small risk of a cancer-causing mutation. An animal with trillions more cells and a lifespan of centuries, like the bowhead whale, should have a vastly higher cumulative risk of cancer. The fact that they do not suggests the evolution of potent cancer resistance mechanisms. The initial investigation sought to determine if bowhead whales employed cancer-resistant strategies similar to those found in other large mammals. The approach was to test these established hypotheses in bowhead whale cells. For instance, elephants possess multiple copies of the tumor suppressor gene TP53. This genetic redundancy leads to a highly sensitive apoptotic response, meaning their cells are quick to undergo programmed cell death upon detecting DNA damage. However, when researchers examined bowhead whale cells, they observed lower basal p53 activity. This was an early indication that the whale utilizes a different biological strategy. This experimental data provides direct evidence. Following exposure to DNA-damaging UVC radiation, bowhead whale fibroblasts did not exhibit a significantly increased rate of apoptosis or necrosis compared to human cells. This confirms that an enhanced cell death response to genotoxic stress is not the primary mechanism of cancer resistance in this species. Further investigation into their cellular biology revealed other unique features. While their cells do undergo replicative senescence like human cells, the phenotype of these senescent cells is different. Specifically, they show a reduced induction of the pro-inflammatory factors associated with the senescence-associated secretory phenotype, or SASP. This attenuated inflammatory profile in senescent cells may contribute to their healthy aging by reducing chronic, low-grade inflammation. The investigation then led to a rather counterintuitive finding. One might expect that whale cells would have more robust safeguards against malignant transformation. However, the opposite appeared to be true, which only deepened the paradox. This table illustrates the minimum number of oncogenic hits, or mutations in key tumor suppressor and oncogenes, required to transform a normal fibroblast into a cancerous one. Mouse cells require only 2. Human cells are more resilient, requiring 5. Contrary to expectations, bowhead whale cells require only 4 hits for transformation, 1 fewer than human cells. This suggests the resistance is not due to a greater number of cellular safeguards. The resolution to this puzzle is found here. Although fewer hits are required for transformation, the rate at which those hits occur is substantially lower. This chart shows the frequency of spontaneous, de novo single nucleotide variants in tumor xenografts. The mutation rate in bowhead whale cells is significantly lower than in human or mouse cells. The strategy, therefore, is not to have more locks, but to maintain a much lower rate of mutation accumulation in the first place. This leads us to the central finding of the research. The bowhead whale's primary defense against cancer and aging is superior genome maintenance, specifically a highly efficient system for repairing DNA double-strand breaks, which are among the most cytotoxic forms of DNA damage. Let's examine the evidence. This graph displays the efficiency of non-homologous end joining, or NHEJ, a major pathway that directly ligates broken DNA ends. As the data shows, whale cells perform this type of repair with significantly greater frequency and efficiency than cells from the other mammals tested. This enhanced capability extends to the second major repair pathway, homologous recombination. HR is a higher fidelity pathway that uses a homologous DNA template to ensure accurate repair. Here again, bowhead whale cells demonstrate a substantially higher efficiency, indicating a superior capacity for both major forms of double-strand break repair. The functional consequence of this enhanced repair is increased genomic stability. This chart measures the formation of micronuclei, which are markers of chromosome breaks and genomic instability, following gamma irradiation. The frequency of micronuclei formation in bowhead whale cells is markedly lower than in human cells, providing a clear visual representation of their genome's resilience to damage. Beyond efficiency, the repair is also more accurate. In this experiment, CRISPR was used to create a targeted double-strand break in the PTEN gene. The chart shows the outcomes of the repair process. For whale cells, a much larger fraction of the repaired alleles are restored perfectly to their original sequence, with fewer insertions and deletions. This demonstrates that their repair machinery is not only efficient, but also remarkably precise. So we've established the what? Highly efficient and accurate DNA repair. The subsequent question is, how? What specific molecular factors are responsible for this enhanced capability? A proteomic and transcriptomic analysis identified a primary candidate, the cold-inducible RNA-binding protein, or CIRB. What made this protein stand out was its marked abundance in bowhead whale cells and tissues compared to other mammals. This high level of expression suggested it could be a key component of their enhanced repair system. To test this hypothesis, a gain-of-function experiment was performed. Bowhead whale CIRBP was overexpressed in human cells. The result was a significant increase in the efficiency of both NHEJ and HR pathways in these human cells. This demonstrated that introducing this single whale protein can confer an enhanced DNA repair capability. The complementary loss-of-function experiment provided further confirmation. When CIRB was depleted from bowhead whale cells using siRNA, their innate, high-efficiency DNA repair capacity was significantly reduced. Taken together, these two experiments establish a direct causal relationship between high CIRB levels and enhanced genome maintenance. So what is the proposed mechanism of action for CIRBP? It appears to act as a crucial facilitator at the site of DNA damage. It is rapidly recruited to double-strand breaks, where it functions as a scaffold, promoting the recruitment of other core repair proteins. It also protects the exposed DNA ends from enzymatic degradation or resection, thereby preserving the genetic information and promoting accurate end joining. We've seen that a single protein in bowhead whales significantly enhances DNA repair. This finding has broader implications, connecting the molecular mechanism to organismal longevity and potentially to human health. To assess the in vivo effects, the bowhead whale CIRBP gene was expressed in fruit flies, a common model for aging research. The results were notable. The flies expressing whale CIRBP not only showed increased resistance to DNA damage, but as this survival curve shows, they also exhibited a significant extension of their median lifespan compared to control flies. This demonstrates that enhancing this pathway can impact longevity at the organismal level. This research culminates in a compelling forward-looking question. The bowhead whale's biology suggests that enhancing endogenous genome maintenance pathways is a viable evolutionary strategy for extending healthspan and lifespan. This naturally leads us to ask, could this principle be translated into therapeutic strategies? Could the targeted enhancement of genome maintenance serve as a novel approach to combat human age-related diseases? The findings presented here provide a strong foundation for exploring that very possibility.